Beautiful talk, thank you very much. I was wondering what's the relationship of the receptors, especially the heat receptors, with the unmyelinated C fibers, which are the chemoreceptors, which also respond to capsaicin. And are there similar transduction mechanisms, or the C fibers, the unmyelinated C fibers that act as chemoreceptors?
Use the microphone. Use the microphone.
The microphone.
The fibers that conduct temperature and also pain are unmyelinated. They're slow fibers, okay? And actually, when you hit your finger with a hammer, okay, you feel two pains, okay? An instantaneous pain, a very hard pain, and then a slow pain. And that's the reason that the two different fibers that conduct, that is the C fibers and the delta fibers that have different conduction velocities. And that's the reason why you feel two pains. Yeah. But those are also temperature sensitive.
What was new to me is that the temperature receptors also respond to capsaicin, which is one of the main stimulants of the C fiber, of the sensory chemoreceptors, the so-called chemoreceptors, like the ones that respond to bradykinin.
Well, that's new to me, but what I thought that was essentially those free terminals, okay, that are unmyelinated fibers that are sensitive to capsaicin, but those are the ones that contain TRPV1, because myelinated fibers don't contain TRPV1.
Do the receptors for pain respond to bradykinin, for example?
I don't know.
Because that's the interface which I'm interested in.
Dr. Luis Herrera Estrella in the back.
Ramon, very nice presentation. I have a very simple question. Do these receptors evolve to sense temperature and they just happen to bind compounds present in food? Or did they evolve to sense compounds present in food and then adapted to sense temperature?
Well, remember that we are in the Vatican, and only that that's an answer that I don't know. What evolved first? I think maybe they're together. What happened is that if the channel, okay, in evolution appeared as a membrane protein containing all the domains that I showed to you, that essentially binding capsaicin and sensing temperature, okay, are located in different domains and they appear at the same time. If the channel evolved that way, but I don't know, maybe it evolved in a different way. You see what I'm trying to say?
Thank you, Ramon. Very nice. I'm still wondering how does temperature, what does a molecular change does temperature produce in the molecule? And this region, the C-terminal, is highly charged, and is it the same in the cold sensing receptor as in the heat sensing receptor?
Yeah. Well, one of the things that you can see in the structures of TRPV1 is the following, that when the channel opens, actually the diameter of the pore changes a lot, but the general structure of the channel doesn't change much, okay? So many people thought, okay, for example, to explain the high temperature sensitivity, that parts of the protein were denaturing, okay? Mm-hmm. But that doesn't happen. You don't see, that 20 amino acids that are hydrophobic are exposed, for example, to the solution. That doesn't happen. So that's the reason why we start using anisotropic thermal diffusion, because actually, as I told you, temperature doesn't distribute homogeneously. It go through allosteric pathway. And so essentially, what we think is that, let me put it in a different way. There are voltage-dependent channels that have the same structure, okay, potassium channels, that open with depolarization and a different kind of channels that open with hyperpolarization. So essentially, the sensor does the same thing, but the coupling is different. In one situation, the coupling will close the channel, and in the other situation, the sensor will open the channel. You see what I'm trying to say?
Yeah, but then the sensor is highly charged. Is that the case?
No.
Amino acid?
What happened, the sensor, I don't know the characteristics. Well, let me see. Maybe I will not be here if I will really know everything about the temperature sensor, okay? But essentially, one thing is that it needs to be exposed to the solution. And both the coil domain in trip main and the membrane terminal and the membrane proximal domain in trip B1 are exposed to the solution. And the only thing that I can tell you is that if you apply thermal energy there, it goes from that place to the gates. That's all.
The next question is by Carlos Bustamante.
Ramon, great talk. I wanted to ask you, you show us that you can calculate by changing the temperature, the enthalpy between the closed and the open states of the channel, the enthalpy change in the process. And so that gives us the sense of how the equilibrium between the closed and the open state actually is regulated by temperature. So my first question is, if you do that experiment in the presence of capsaicin or one of menthol or whatever, do you actually can see a change in the enthalpy, in other words, in the sensitivity of the channel to the change in temperature?
Okay.
That's my first question. I have two questions. No, but answer this, please.
What happened essentially is that, okay, you can model the whole thing with an allosteric model. Essentially, assuming that capsaicin is binding to one place, and you have the temperature sensor in a different place, and they couple together as allosteric factors. Okay, so if you assume that what you are going and what you actually found, okay, is that the probability of opening with temperature, let's say, has a threshold in a certain place. I mean, when you are changing capsaicin, okay. You shift, okay, the probability of opening versus capsaicin towards the right, towards the left, if you increase the temperature in the case of 3B1. And that is perfectly explainable by an allosteric model. Actually, we model TRIP-MA, okay, temperature and voltage with a simple allosteric model, and that allosteric model explains everything. So essentially, if you activate one temperature sensor, okay, now it's more easy to activate the channel if you activate the voltage sensor. You see what I'm trying to say?
Yes, I see.
And they're coupled by allosteric factors.
So it's like an allosteric control, and you can actually have, and your T1/2, which would be the temperature which you have is 50% open and 50% closed, is shifted.
One nice thing that I didn't tell you is that you can have all other sensors addressed, and you can activate the channel only with temperature. Okay. So essentially, it should be a temperature sensor somewhere in the channel.
Yeah. My second question quickly is, you were able to use molecular dynamics simulations to try to look at the path that the heat may actually take in order to transfer from the sensor itself to the channel itself.
Right.
I was wondering if it would be possible to do an experiment where you put a fluorophore somewhere in a place where you predict the heat will pass, and then excite optically and sink heat, essentially in the form of absorbed temperature, absorbed energy, and see if that actually activates the channel.
We haven't done that, but I can tell you what we are doing now. I mean, you can do actually an experimental anisotropical thermal diffusion. And what we are doing essentially is using gold particles. Gold particles can be of 20 Armstrong in diameter. And if you heat them with visible light, you get heat. So you can transmit heat to a very small region of the protein. And you can bind the gold particle to any place you want in the channel. So that's the way that we think we are going to
That's
do.
another way, yes.
I have a question. Could you comment on the heat receptor of the pit viper snakes?
The what?
Heat receptors of the rattlesnake snakes.
Well, that is an infrared receptor. And what happened in that case, in the head of the snail, of the rattlesnake, and in serpents in general, okay, they have these organs that are called the pit organs. And it's a different receptor. What happened is that TRPA1 can be a cold receptor and can be a heat receptor. In the case of the snake, it's a heat receptor. And in the pit in the pit, you have, a huge expression of TRPA1. So actually, when the snake is going around, it's sensing heat about 1 meter. It can distinguish a change in temperature 1 degree. So essentially, that's the way, yeah. But it's a heat receptor, and it's TRPA1, it's not TRPB1. Bats use the vampiros, no, the vampires use TRPB1 as infrared receptors. And what is more amazing, okay, is one mutation in camels and squirrels allow them to support 50 degrees with no pain.